Yeah, so good morning, everybody. Welcome to today's work in progress seminar. Today we have Lydia Fyie, who's going to be who's a new postdoc research fellow working with Ali Gardner at the University of Maine. She received her BA in biology and theater arts from Kalamazoo College, liberal arts in Kalamazoo, Michigan. She earned her PhD the spring of 2024 in entomology from Ohio State University, studying the effects of urbanization on mosquito dormancy, co-advised by Megan Muti and Mary Gardner. Her research focuses on how human-mediated ecosystems alter the ecophysiology of arthropods and how we can use that information to understand vector ecology. Aside from science, Lydia is an avid supporter of the performing arts and continues to participate in local theaters throughout her PhD career. So, with that said, she's going to be talking about some of the work that she's done and some of the work that she will be doing. So, I'll open up the floor to her.
Right, yeah, thank you so much. Like Nolan said, I'm just started, so I'm going to be sharing some stuff about what I did at Ohio State, and then starting to talk about what I'm planning on doing here in Maine. Just because there's not that much to share at this point, aside from the design elements, the plan as it is. So let's see if that works. All right. Okay, so talking about seasonal environmental change and vector ecophysiology, mentioned going to be talking first about what my dissertation research was about, which was broadly how changes in seasonal cues and in urban environments alters dormancy in the northern house mosquito. And then we'll move on to talking about what my postdoc research is going to be, which broadly be looking at things about the mechanisms and environmental factors influencing overwintering in the black-legged tick in Maine. So first I'll start. Some of this is just some overview of my system for people who may not be as super familiar with urban ecology as I am. So urban systems have a lot of different environmental factors that could be affecting insects and other arthropods in a lot of different ways. I was mostly focusing on heat, so temperature and light. So for temperature, this is known as the urban heat island effect. This is the um, phenomenon where cities or any area with high human settlement, so this can also happen in towns, but to a lesser degree, um, that area is going to be warmer on average than the surrounding areas. And depending on season, time of day, and also the surrounding biome, so the climate that you're in, this can be anywhere from 1.5 degrees warmer to, in some situations, recorded to be almost 10 degrees warmer. So it can be pretty intense. There we go. So yeah, it goes up and the temperature also increases. The other thing I was really interested in was how cities can have altered light regimes. So during the night, we have a situation where all of these different structures, like our streetlights and the lights on the buildings or inside the buildings, create an overall brightening or light pollution caused by artificial light at night or ALAN, where the ambient light in cities can be as bright as zero point 0.1 to even 4 lux brighter even when you're under direct illumination. So right there, I have that like a clear starry night is has a little bit of light, but much at a lower degree than from light pollution. And this is a problem because both temperature and light are cues that organisms use in order to determine the season and then alter not only daily, but also seasonal biology. Um, so forward one. So, as I said, the reason that this is important is because both day length or photo period and temperature, but mostly focusing on day length here because day length is one of the primary cues used in temperate ecosystems as an environmental cue for seasonal timing. The reason of this is that day length has been an extremely stable environmental thing for millennia, although a winter or a fall may be warmer or cooler depending on the year. The photo period is going to be basically the same. So this allows organisms to synchronize their development with favorable conditions. To demonstrate this, I'm going to just sort of walk through the dormancy of my study organism, which was the northern house mosquito or Culex pipiens. So adult females of the species enter a facultative diapause cued by day length and temperature. And luckily for me, there are very specific phenotypes and behaviors that are associated with diapause, which makes it pretty clear to tell when they're dormant and when they're not. So under long day conditions, when they also have higher temperatures, mosquitoes are investing their energy into reproduction. So you can see this, they will have these large egg follicles right here when you dissect out the ovaries and you can measure them. They also have low nutrient reserves. So if you're looking at their total lipid content, it's going to be lower. And behaviorally, they're actively host seeking and blood feeding and thus reproducing and able to, of course, spread pathogens. However, in short day conditions, so depending on where you are on the latitude when this happens will change. But at some point, days get short enough and the temperatures get lower so that they initiate diapause. So they are shifting their energy towards accumulating those research reserves and halting their reproductive development. So you will dissect out their ovaries. They have egg follicles that are halted at an earlier developmental stage. And also they will have, of course, much higher lipid reserves. And then behaviorally, and for in interest of vector-borne disease, they halt blood feeding, instead seeking out sheltered hibernacula to survive the incoming inclement conditions. My general hypothesis was that Allen and urban warming both had the potential to inhibit this response by essentially tricking the mosquito into thinking that it was still in early fall or in those good summer-like conditions rather than heading into November. So the effects of Allen on diapause has been shown in several different moth species, flesh flies, and also the tiger mosquito, Aedes albopictus. So this appears to be somewhat widespread of a possible impact. But I overall was looking at whether urban environmental disturbances inhibited diapause initiation in Culex pipiens, so both urban heat island and Allen. And I did this by first looking at both of these in a lab setting, so doing a laboratory experiment. And then I went out into the field to conduct a semi-field experiment that actually was originally designed to just look at Allen, but we actually got some pretty good spread on temperature as well. So we were able to start teasing apart the relative impacts of both of these. So for my laboratory experiment, this is two different ones, but I've put them together because the design is very, very similar. So for both of them, we had our mosquitoes reared in either a diapause-inducing control, so something that we knew that those conditions, that temperature and that photoperiod would get them in diapause regularly. And then they were either in the same conditions, either with a pretty low level of Allen, or we reared them at 2 additional temperatures with an increase of 2 degrees or an increase of 4 degrees Celsius, which was based on some previously published data to say that this was what the urban heat island would be around in the fall for the Midwest region that Ohio is located in. And then so they were reared all the way to adulthood, and then I checked their the phenotypes that are commonly associated with diapause. So we looked at their lipid content using vanillin phosphoric acid assay, dissected out their ovaries and measured the average egg follicle size in each individual. And then also I measured their blood feeding proclivity. So I offered them a blood meal, counted how many actually took that meal. And yeah, so what did I see? Oh yeah, there's just for as a reminder what we would expect if they had actually averted diapause, we would see that they would have lower lipid content, but higher egg follicles and larger egg follicles and a higher blood feeding proclivity. So we found, first off, that only exposure to alum actually reduced lipid content. So here we have just the lipid content in an individual sample from the mosquito normalized to the mosquito's wet weight. And our alum exposed individuals had significantly less, although it wasn't like a huge reduction. But both the 22 degrees and the 24 degrees had no change in their overall total lipid content. However, both alum and those increased temperatures did increase egg follicle size, and they all also increased blood feeding. So based on that, we can say that both of these both both of these different factors have the potential to cause them to avert diapause in that they are reproductively active. However, there seems to be some kind of decoupling, at least with the temperature, of that change in also increasing their nutrient reserves. Up here on these, we have the average egg follicle size here. This percentage is how many females would have been considered in diapause based on their egg follicle size. So interest, we had very high in the diapause control, as we would expect, but when they were exposed to alum, only 18% of the female mosquitoes would be in diapause. So that's pretty low, a pretty significant impact. And then at 24 degrees, it was 39. So these females are actively biting, and then they are actively reproducing, because I did also offer blood-fed mosquitoes oviposition water, and we did get egg rafts that did hatch from those that fed. So I'll go straight forward. So from there, and this was a question I got all the time, was whether these would actually still impact them in the field. Those were reared in growth chambers, so it was white, controlled, not exposed to the same like variation in temperature that you would get actually outside. So wanted to see whether or not we could actually still see this when they were put outdoors. To do this, I reared mosquitoes in these little butterfly cages, either exposed directly to a light at night. So this is a porch light right here. So it's again completely there. And then in the same area, there was a relatively dark spot that the mosquitoes were also in. So it was a paired design in that way. Then ran this experiment both in 2022 and then in 2023, adding September in 2023, just to see if we could see any difference between month, because obviously day length is going to be pretty different between those two. And so we may see that at some point in the year, it gets too late that even if there is an effect of either of these, you're not going to see it anymore. So mosquitoes were deployed as first instar larvae inside these beautifully made emergence jars that I didn't want to have to buy the actual ones from WIP or anything. So I made these myself with jars and plastic cut funnels. And then they were reared to adulthood until they were approximately a week old, or until the first hard freeze of the year, in which they were pulled back into the lab. To account for other microclimate variables that may influence diapause, I also installed temperature data loggers inside each enclosure, which also recorded light levels as well. So I was then able to also go out at night one time, take a handheld measurement. So I was able to get both the average daily temperature at each location as well as the nighttime brightness as a measure of all of the different light variables, including the average across the entire period, that one-time measurement, as well as like just how bright it got at night and also the minimum. As before, measured their lipid content, dissected egg follicles, and then also from there categorized mosquitoes into whether they were in diapause or not to get the diapause incidence per enclosure, as well as their percent blood feeding again. And then also, as before, took those blood-fed mosquitoes, gave them the opportunity to actually lay eggs, and then recorded how many females did actually lay egg grafts, and then see how many of those egg grafts actually hatched any larvae at all. So found some pretty interesting stuff. So that's just how we did. We took all, as I mentioned, as I sort of alluded to, we took all of those nighttime brightness variables and put them into a principal component, extracted the first, no, sorry, principal component analysis, PCA, extracted the first principal component to get a measure of the nighttime brightness for each site. So found some pretty interesting stuff. So here are the results for September. So the asterisk at the top indicates that the interaction between night brightness and the average daily temperature was significant. And then here with this little star here that says that the slope of that line is significant. So what we saw is that first egg follicle size, there was a significant interaction between allen and warming. However, what I think is most interesting is that it appears across the board, allen inhibited diapause while also suppressing the effects of warming. So in our dark individuals, so these two lines are holding night brightness at the value for median value for our allen sites or our dark sites. So this dashed line are the dark individuals. So we see that there is a pretty significant effect of average daily temperature for those that were reared in the dark. However, that effect kind of just completely melts away under those that were exposed to allen. We see something that's basically the same when you just look at diapause probability. So diapause probability goes down as temperature increases for the dark sites, but there is no such effect for allen. For blood feeding probability, again, this one's pretty stark as temperature increases for the dark sites. You see that their blood feeding probability goes up, but when they were exposed to allen in September, they had consistently high levels of blood feeding the whole way. And then I saw no effect of either on lipid content when they were in the field. And then in October, we saw that the effect of allen continues into late fall. So not only is allen obliterating the effect of warming in September, we still see it having an effect into October. And these mosquitoes were emerging in like the beginning of November, which is pretty significant for them to still be in any way reproductively active, possibly at that time. So again, see that there's a significant effect of night brightness and average daily temperature for egg follicle size and diapause probability. For blood feeding, it was only a main effect of night brightness. So although it was very like low levels of blood feeding among those exposed to light, they still did actually feed in any capacity, which they did not do when they were in the dark. In the dark, we had 100% diapause incidence, so not a single one blood fed. And then among those that did blood feed, we did see that they were also able to produce offspring. So in September, we obviously, if they were in the dark and they blood fed, then that just means that they were in natural levels of variation. However, if they were exposed to Allen, they also did actually produce egg grafts, and pretty high numbers of those egg grafts actually did hatch. However, interest in October in the dark conditions, not a single mosquito took a blood meal, so we got no egg grafts, no larvae at all. But when they did blood feed, when they were exposed to Allen in October, pretty high numbers actually did go ahead and actually lay an egg graft, and those egg grafts seem to actually hatch. So in theory, this means that if mosquitoes, at least in Columbus, Ohio, are under a light source, in the larvae are under a light source in October, it seems possible that they could be emerging as adults that are still reproductively active. And if the conditions are right, they could take a blood meal and produce more larvae. And if conditions continue to stay right, we could see a longer time frame when mosquitoes are active into late fall. So this leads me to what I am planning to do for my postdoctoral research plans. So I have been interested in both seasonal biology and in thermal ecophysiology for some time. And one question that I have had here that's once I got here in Maine that I have been interested in is this question of whether winter cold can limit the northern expansion of ticks, including Ixodes scapularis. One thing to point out, the overwintering of arthropods in general in changing climates can have some pretty significant implication for population dynamics. And in part, it is important to think about winter because winter is a season that appears to be having quite significant changes in temperature and like precipitation as a result of climate change. And also as a result of other forms of environmental change, whether they be coming from humans or not. So that's why I wanted to focus seasonally on what is happening in winter. This past work has found that black-legged tick does have generally high overwintering survival under natural conditions. However, it is important to note that both abiotic and biotic factors can shift overwintering mortality, making them more likely to experience high mortality or not. Depending on which study you're looking at, the removal of both snow and leaf litter can significantly alter overwintering survival, perhaps by changing the level of insulation that the ticks are receiving. So they could be warmer under the leaf litter, so you could, and the snow, so you could have that. Removing that would then make them actually be exposed to the cold, or you could have changes in humidity, any such thing. Past work in the Gardner lab did find that both climate across a large latitude gradient as well as the presence of snow did significantly impact tick overwintering survival in Maine. So I wanted to start drilling a little bit more into the mechanisms and some other abiotic factors that could alter their overwintering success. So some of the main questions that I wanted to address was, one, what is a probable mechanism for their overwintering mortality? Would that be direct cold injury or the premature depletion of nutrient reserves? There is some evidence that mortality is higher in the spring due to increases in energy usage. However, this particular aspect has not been looked at as much when you get into the more northern climates, where they could be theoretically exposed to more intense cold. They also wanted to look at weather insulation from different leaf litter types. So I'm focusing on coniferous versus deciduous, just say broad group. Can that alter their overwintering mortality? And then also whether the impact of snow interacts with this leaf litter insulation. So theoretically, leaf litter could have no effect when there's snow on top of, like pretty significant snow on top of them. But if you take that snow away, then all they have is the leaf litter, and then that could be a problem. And then I have some secondary questions on based on how the experimental design has turned out. So first, whether overwintering mortality and energy usage differs between the nymphs and the adult ticks, as well as whether we can also see higher survival in infected ticks. So there is evidence that infection with Borrelia does increase overwintering survival, although it did not affect activity levels or cold tolerance. So how exactly that's happening needs to be investigated a little bit more. But obviously, that's not the only pathogen that ticks can be infected with. So whether or not something like anaplasma infection or co-infection also alters survival should be pretty interesting. So this is just a diagram of what the experiment is going to be. I am going to be literally setting this up this week. So on Friday, all the ticks should be going out in the ground for this first year. So all I have is this diagram. I don't have pictures yet. So first, going to they're doing here in central Maine for a climate area, but I hope to add northern Maine in next winter so that we can get an idea of different large scale climate differences. This is a factorial design, so as we go down, going to keep these different groups. Then we'll have forest type or leaf litter type. So I have different stands in a managed forest at the University of Maine, which are either softwood or hardwood stands. And then within each of those, we also have insulation manipulation. So whether we allow snow to just accumulate on top of the enclosures, or we remove the snow after each major precipitation event. So then ticks will be enclosed into tubes, and then those tubes put inside buckets filled with soil, which are then placed into the ground and covered with the leaf litter or snow or without snow. We'll also be measuring the microclimate at the leaf litter level, so getting their temperature and also relative humidity and tracking that between all of our treatments. Then I have some field collected ticks. So I got a bunch of adults this past month from surrounding woodland areas here in Maine. I also have some lab colony nymphs from the Oklahoma State tick rearing facility. So ticks will be placed in tubes, and I'm going to go out planning, hopefully, we'll see how this works, doing a biweekly check of their survival so that I can monitor over time their mortality and maybe check and see at what point do we actually see the largest die off. So when across the winter are we actually seeing mortality? Depending, even if they survive or if they die, still we'll check the infection status so that we get those data, because as I mentioned, there is evidence that at least Borrelia infection does alter their survival. So that seems pretty important to at least control for. I also am planning to use a assay that allows you to measure the total protein, total carbohydrate, glycogen, and total lipids using colorimetric assays. So like the Bradford assay for protein, same vanillin assay that I use in the mosquitoes for total lipids, and then anthrone for carbohydrates and glycogen. And you can do this in one organism. So a single tick, you can get all of these, and that allows you to measure something of the complete energy budget for that individual. So I'm planning on measuring that both in the survivors that of ticks that survive all the way to spring, as well as those that have died. So we can see maybe they've completely gone through or they have significantly depleted one of these reserves at different times. I also, for the lab colony nymphs, have a group that we're going to take out monthly and measure that complete energy budget. So this is just one aspect of what I'm thinking of doing. So some additional possible research avenues to look into a little bit more. First, after this season, I want to first off examine my preliminary evidence from this winter on what the source of mortality actually may be. So some other things that it could be is that there is sublethal cold exposure. So they're not dying from directly from the cold. So it's not like they're freezing and then, or they're getting too low below their thermal minimum and then dying from that, but they still could get cold enough that they're accumulating cold injury or something else that then causes injury that they have to repair or other kinds of stress responses that are altering their ability to survive as long. So that's something though to chew on and think about how I can start like teasing that apart. There is also additional environmental stressors that are present in managed and human-mediated ecosystems that could be altering their mortality, both from those stress or immune responses or anything else. I also want to actually verify the cold tolerance in field collected ticks. This has been done before from different populations. However, I suspect that there could be some differences based on population, so latitude or other kinds of landscape gradients, meaning that these ticks are adapted to a specific area. Also, one question that I have is whether the fall conditions allow the ticks to acclimate better. So if you have ticks that are up in a more northern climate, they acclimating to those fall conditions, allowing them to survive better up there versus if they were down in a more southern climate. And all that can help us be able to predict whether they can actually survive in these as ticks spread north, or is the changing climate going to possibly start to provide more limitations? Another thing I want to do is start to leverage some long-term passive surveillance data from public tick submissions to UMaine Extension to evaluate if changes in winter temperatures contributed to changes in tick phenology primarily. I'm obviously very interested in what's happening in both fall and winter, just because those are less examined aspects of seasonal biology. And I think given the large scale changes in both of those seasons, as a result of climate change, it is pretty important to see what possible impacts we could have. And with that, I'll just open to any questions that we might have. Yeah, it's only 30 minutes, but it's what I got for today.
Thank you, Lydia. So I'll open the floor to any questions anyone wants to ask. Feel free to unmute or type it in the chat.
Yeah, go ahead.
Me?
Yeah.
Did I misunderstood must hear, or did you say there were overwintering nymphs in Maine?
Mm-hmm.
How do you infer that? How do you know they're, what does that look like?
Oh, I'm like what my experiment is, or that word. I'm sorry, I don't quite understand what you're asking.
Okay, so the nymphal season here is in late spring, summer,
Yeah.
and it's usually over by late summer.
Hmm.
So if overwintering nymphs were to appear, would they appear in advance of the regular nymph season or during the nymph season? And if so, how would you know they had overwintered?
Um, I don't know that.
Okay.
What I do know is that, um, like a lot of the overwintering studies have used nymphs. I am assuming that's because of their like epidemiological importance and also just ease of access to them. So the like that is fair that the field relevance of overwinter of the nymphs in winter may be different than the adults, but um like yeah, I don't know about that specifically. All right, another hand up.
Andrew, go ahead.
Yeah, I'm just curious about the Helix pipiens. Is there any reason to think there's no way of knowing, but is that likely a general phenomena with the exposure to ALAN giving them a longer period for a lot of different genera?
I suspect that it is.
Mhm.
Um, like I mentioned, this has been seen in moths, so it's there in Lepidoptera, it's there
Mhm.
in well, other dipterans, so flesh
Yeah,
flies as well. There's someone in Japan actually who did an experiment with flesh flies, um, and saw the same thing. And it's happening in like pupal diapause, obviously adults here, Aedes albopictus, so embryonic. So it,
yeah.
it does appear like it's
That's a
pretty
pretty
widespread.
yeah. So maybe a lot of the effects we think we're seeing from temperature dependent climate change are actually modulated by light exposure.
Yeah,
And
that's
that's it's yeah, it's that interaction term is really interesting.
yeah, it was. One thing to keep in mind is that um especially when you have them in a mobile life stage,
Mhm.
they may be escaping the light. Like the person I mentioned in Japan, I was talking with him at ICE about it, and he mentioned like he had a thing where like the flesh flies outside would just leave. They would just go somewhere else away from the light source. Um,
I
that's
see.
why with the mosquitoes, it's more interesting because they are completely locked in one spot.
Yeah, yeah.
Um,
And like direct exposure. All
yeah.
right, cool. All right, thanks.
Go ahead, Lars.
Yeah, I was just wondering for the overwintering experiment, if you
Mhm.
are checking the ticks that are placed in the tubes every couple of weeks, how will you know if they're alive or dead? Especially
Um,
if they are in diapause.
Planning on allowing them to warm up and see if they are then actively moving or not. That would be what we would do if we brought them back into the lab at the end of the field season anyway. Um, there is a possibility that doing that is going to cause them to go through more variation in temperature and increase mortality there. But like it's kind of a trade-off in how to get that information of like mortality across time. Um, so this is something I have thought about pretty intensely about what the best way to go about getting that information would be.
Any other questions? So if that's it, we'll end the meeting now or the seminar now. And feel free to reach out to me or Lydia if you have any questions on our work. Thank you again, Lydia. And all right, take care, everybody.